All right, welcome everyone to session 2 of the NADP clad Critical Loads of Atmospheric Deposition seminar series. Really excited today to have two great speakers from the National Forest Service who are going to be talking to us about critical loads of nitrogen and sulfur for epiphytic macrolichens. My name is Mike Bell. I'm a biologist with the National Park Service Air Resources Division and a member of the NADP Critical Loads of Atmospheric Deposition team and have been helping to put together this seminar series. For those of you who weren't here last week, just wanted to highlight this is or last month, this is a 7-part seminar series where last month we went through the kind of a general overview of critical loads and how we evaluate how air pollution is affecting different ecosystem processes. And then over the next 6 months, we're going to be diving into individual ecosystem components and really understanding what is currently known and unknown about those responses. So we hope you can join us for all of the subsequent sessions. Just to give a quick overview, the NADP, or the National Atmospheric Deposition Program, is based out of the Wisconsin State Lab of Hygiene in Madison, Wisconsin. The NADP has been monitoring deposition of air pollutants for 43 years using 5 national networks, the largest of which has 263 sites across the US. There's also 4 science committees, of which CLAD is one, to help translate the data that we're getting through the monitoring into applicable science outputs. If you're interested in getting more information on NADP, there is the link at the bottom of the page. Specifically, CLAD is the Critical Loads of Atmospheric Deposition Science Committee. The goal of our group is to facilitate technical information sharing on critical load topics with a broad multi-agency and entity audience. Our goal is to fill gaps around the thresholds of air pollution at which change is occurring in ecosystems and provide consistency in development and use of these critical load thresholds across the US. We also are trying, through efforts like this seminar series, to promote the understanding of critical loads and how they're applicable to land management and policy. Generally, we have 2 main meetings at the NADP spring and fall meetings. The spring meeting, which will be, I believe, the week of May 10th, we'll be congregating and talking about all the different critical load projects currently occurring. I also want to mention at the end of the meeting, there will be a brief seminar, just like last time, kind of highlighting what worked, what didn't work in these presentations, what more would you like from future presenters, so we can make sure we're giving you what you need, what you're most interested in. So with that, I'm going to stop sharing and hand this over to Linda Geiser. So Linda Geiser, Linda, I'm assuming you're starting this off, I guess?
Actually, Rob's going to start us off and I'm going to finish us off.
Okay, well then let me introduce Rob Smith. Rob Smith is a forest ecologist and ORISE postdoctoral fellow with the U.S. Forest Service Air Resource Management Program out of Washington, D.C. He was previously a postdoctoral research ecologist with the Forest Service, forest inventory and analysis in the Pacific Northwest, and earned his PhD in teaching at Oregon State University. Currently with the AIR program, he's developing reporting and mapping tools that let managers plan for vegetation responses to pressing global changes. Rob, thanks for being here, and you can share your screen.
Well, thank you very much. I will share the screen now. All right, hopefully we have a full screen presentation about to pop up here. Thanks, Mike, for the introduction, and it's great to have the opportunity to share with you all today about lichen-based critical loads for U.S. forests. So Linda probably does deserve an introduction, but I'll let Mike do that, I guess, when I turn it over. But I will just as a general overview, what we hope to do today between Linda and myself is to share with you why we care about monitoring lichens and some of the ecosystem functions that they perform. We will look at how we actually arrive at a lichen-based critical load using something called airscores, among other metrics, and also how we quantify uncertainty around the critical loads and around exceedances of those critical loads. And then, so I'll talk broadly about those first two issues, and Linda will cover more of the applications to answering managers' questions and showing applications for federal agency uses. I do also welcome questions at any time, but you might have to, maybe I'll let Mike route any of those to me. So kind of to get that first question, why do we monitor lichens? There's a few reasons. One is that they're extremely sensitive to atmospheric deposition. They don't have roots, they don't have a waxy cuticle, they don't have bark like a tree might have, so they're directly exposed to the atmosphere, unlike the trees and woody shrubs that they grow harmlessly upon the outside of. So directly exposed to the atmosphere means they depend on it for their nutrition and water relations. And that also means that they are decoupled from any soil or terrestrial processes. So responses in lichen communities should be directly reflective of atmospheric changes, much more so than the plants that they grow on, or at least respond on a shorter time interval. So in essence, where the idea of monitoring lichens is to be the canary in the coal mine for atmospheric changes. We take a community-based approach, which we find more informative than measuring single species. So for example, as you proceed along a deposition gradient from lower to higher deposition, perhaps unsurprisingly, you get turnover in the kinds of species that you would observe. So you have mainly sensitive species at the low end of a deposition gradient, turning over through moderate and then extremely tolerant species at the high end of that gradient. So if we were only to measure a single species, let's say we focused on only a sensitive species, by the time that that species disappeared midway along the gradient, it would no longer be able to provide information about deposition at higher levels along that gradient. So the community approach gives us richer information than any single species alone. Another reason we're interested, I mentioned they're sensitive, but importantly, lichens are sensitive at fairly low levels of deposition. So if we were on a deposition gradient, run it increasing from left to right along this lower axis, lichens among all other kinds of organisms and ecosystem processes are change at relatively low levels of deposition. And in fact, only diatoms and some aquatic indicators might be indicative of lower changes in deposition. This is, for example, on a nitrogen gradient, although it's also true for sulfur. And finally, one more reason that we're interested in monitoring lichens is to leverage an information-rich nationwide data set. Over 10,000 plots measured in a nationally consistent way by the Forest Inventory and Analysis Program and the AIR program using an identical measurement protocol that gives us, for each of these plots, a complete species inventory. So if we know the community of species at each of these sites, and we are covering such a large geographic area, another benefit is that we're covering a large gradient in nitrogen and sulfur deposition. So we can use all of this information to tell us something about how lichen responses may reflect atmospheric deposition. So I want to diverge briefly and just talk about what is it that lichens do and why should we care about them, and talk about ecosystem functions. So if you're a person who's only focused on timber or understory vegetation, you may not be so interested yourself in lichens, but they're doing really important things that impact on other ecosystem components, on other vegetation and other processes. So I like to think of lichens in a sense as a kind of forest fertilizer in nitrogen poor areas. They contribute nitrogen fixation, converting atmospheric nitrogen into biologically usable forms. They serve in a sense as forest air conditioning because they regulate understory canopy thermal budgets by the uptake and release of water, controlling evapotranspiration. They serve as a forest sponge by moderating rates of water uptake and release through the forest ecosystem. And they also serve important roles in interaction with animals and wildlife, serving as important food sources and habitat for threatened and endangered species, deer and other ungulates, mammals, invertebrates, and so forth. So they're doing a lot of important things. And based on these functions, we can categorize them into broad ecosystem functional groups. So we measure critical loads for each of these three functional groups and all groups in total, or all species in total. So I mentioned forage lichens that are important, especially in winter for deer and for mammals and other small mammals. So these often include the hair lichens or the beard lichens. The cyanobacterial lichens are important for performing that nitrogen fixation that contributes nutrients to the system and serves as a nutrient-rich food. These also tend to be really good indicators of forest successional status, forest continuity. And then the catch-all group that's not forage or cyanobacterial lichens is called matrix lichens, which perform a variety of functions, everything from nesting and food materials for animals, and even many human uses, including dye materials and pharmacological materials. So they're doing lots of important things, which makes it useful to break down critical loads for each of these functional groups. So I mentioned forage lichens being the hair or the beard lichens, especially important for winter browse and forage. Cyanolichens having a cyanobacterial or a blue-green algal partner that contributes nitrogen to the forest. And matrix lichens doing, again, an array of things, including some areas where they serve as important nesting materials for birds that then go on to reduce insect populations and increase pollination services that positively benefit economies that are centered on timber or fruit crops or floral crops in a lot of rural areas. So in that sense, lichens are foundational to a lot of the other ecosystem services that other organisms are providing. So that's a little bit of the background about why we should care about lichens. So how do we actually arrive at a lichen critical load? So just as a refresher, as was covered last month, a critical load, if we think about some ecosystem response here on our vertical axis, let's say we have a lichen response that as deposition increases from left to right, we see a change in that response from a relatively better state to a relatively poor state. And the threshold at below which no harmful effects occur is our critical load. So that's the deposition value there, and any other values beyond that are in exceedance of that critical load. So the gap that's between any observed deposition value and the critical load is termed the exceedance. So we're interested in lichen critical loads and lichen exceedances. So I mentioned a lichen response. I should tell you what lichen responses we measure. And they fall into two broad categories, one being species richness or abundance. So we're considering either the count or the abundance of different species belonging to various functional groups that I mentioned earlier, either all species in total, ones that are relatively deposition sensitive, the cyanolichen group, and the forage lichen group. So each of those is giving us a different piece of information about lichen responses in terms of diversity. But then another broad group or another broad lichen response concerns the composition of species that are at a given site. And with that, we use a metric called an airscore which I'll show you in just a moment how we actually measure that and what that means. But it's talking about the relative balance between a community dominated by very sensitive, deposition sensitive species versus very deposition tolerant species. So I'll show you that in a moment. So for these two broad lichen responses, we have two different kinds of critical load model. For the richness models, as we proceed along a deposition gradient, again increasing from left to right, lichen richness for all the functional groups tends to decline. So we decline from having many species to fewer species along that gradient. And we define the lichen critical load for diversity as the point at which you observe a 20% decrease from the peak possible diversity. So what this critical load is telling you is where we observe a harmful decline in species diversity. Whereas the composition metric using the airscores are measuring a critical load where we see no relative change in that airscore. And what that means is low airscores mean that we're dominated by relatively sensitive or clean air species, and higher airscores are dominated by a mixture of more tolerant species. So the place on the gradient where we see that changeover from sensitive to tolerant is what we define as the critical load. And I should mention you're seeing these rainbow colors here, a regression line for different temperatures. What I'm not showing you here is that climate is a covariate that's explicitly included in each of our critical loads models. The critical loads for lichens apply nationwide. For each kind of lichen metric, there's one value that's uniform nationwide. There are places where we cannot make inference about the critical load. So we don't wish to make an estimate in areas that don't have permissible climates or available forest lands because we're measuring epiphytes that grow upon trees and shrubs in forest lands. So within the critical load mapper tool, part of the National Critical Loads database, we have estimates for lichen critical loads, but they are masked by permissible climates and permissible forest lands. So I mentioned each lichen metric has its own single value critical load for both nitrogen and sulfur. Each of those 5 kinds of metrics, the 4 diversity metrics and the community composition metric. I keep talking about this community composition metric, which we've also called the airscore, that talks about community compositional shifts. And I'd like to explain a little bit about what is a lichen airscore. So as I mentioned, we measure species, but we aggregate it to a community level. But each species has a sweet spot on the deposition gradient, an optimum value where it occurs most frequently. So for example, here are 5 different species. These top 2 species with the cool colors have relatively low optimum values on the deposition gradient. Here I keep showing you nitrogen, but the concept is similar for sulfur. And then these hotter color guys have relatively higher tolerances or higher optimum values on that deposition gradient. Again, I don't care about single species, but if I have a site where certain collections of species occur, we can average this optimum value for those species at that site and then call that an airscore. So an airscore is the average of all species optima for that particular community. So for example, if I had site A dominated by 3 sensitive species, the average value of the airscore, the average of these 3 arrows, would be fairly low on this gradient. So that would be a relatively low airscore for site A, versus an alternative site B might be dominated by 3 species that have fairly high tolerances and a fairly high airscore. So Along that gradient, the air score is showing you, as the lichens are expressing it, where you are on the deposition gradient. And we can take these air scores and just map those wherever we have lichen information. And we find that lichen air scores tend to track deposition pretty faithfully. So I'm showing you here a map of the US where each point is a FIA or air program lichen plot, colored by its nitrogen air score in this case, where we see hot spots of higher air scores in perhaps expected areas on the eastern seaboard, in the Central Valley and western Sierra Slope of California. And you can even pick up more subtle patterns like the Puget-Willamette Trough of the Pacific Northwest, dominated by urban and agricultural areas and other places across the nation. So these air scores are important because they are essentially showing you how the lichen communities are observing deposition, if you want to think of it that way. It's the expression of deposition as seen by the lichen communities themselves. Because we're covering such a large geographic area, there's a lot of other things happening that impact lichen communities other than deposition. So obviously across the continental US, there's a variety of climates and forest types and different possible lichen species that could enter any given site. So what we'd like to do is account for some of those things, such as climate being one of the strongest signals. We want to subtract the climate signal from the deposition signal. So where I'm showing you air scores on this vertical axis along the deposition gradient, we find that hotter, drier sites tend to have relatively inflated scores, and cooler, moister sites tend to have lower scores. So we use regression methods, statistical methods, to essentially account for that and downweight those hotter, drier sites and upweight the cool, moist sites, so that at the end of the day, these adjusted air scores much more faithfully represent the deposition gradient. And if that sounds like a lot of, I don't know, statistical nonsense, or why are we doing this sort of thing, the implication here is that at the end of the day, these nationwide critical loads do not depend on the climate or on which species are available at any site. They can apply nationwide because we've already accounted for these things. One question that folks may have is, how much trust should we place in lichen critical loads or exceedances? What is the uncertainty that we have around critical loads? So we can do lichen critical load uncertainty estimation using a bootstrap process, which essentially says, you're giving me one estimate, but please also give me the range of possible outcomes if I had observed a different combination of species. So for each site, each of those 10,000 odd sites across the US, we have the point estimate, that'd be one single point on this plot, but we're also interested in the range of variability around that point. So if I have a relatively narrow bootstrap confidence interval, that means I have a pretty good ability to pinpoint that estimate, so low uncertainty. And by contrast, if these bars are really wide, then I have less ability to pinpoint that estimate, and I have higher uncertainty. So what this looks like, let's say we're interested in using air scores to estimate critical loads exceedances across the US based on lichen critical loads. The exceedances, again, we'd find hotspots in the mid-Atlantic here, California, etc. This is the point estimate, but the patterns of uncertainty across this area actually differ quite a bit. What I'm showing you here are the hotter colors mean higher uncertainty of the estimate, and they're not found where we have higher exceedances. They're actually found in areas where we tend to have fewer lichen species within any of these single plots. So what that means is that patterns of exceedance are not directly related to uncertainty about that exceedance. It's related to other things like species richness. So that naturally led us to the question, well, what if we had captured a different number of species per plot? Did we sample enough per plot? So again, using some bootstrap resampling methods where instead of changing or varying the potential combination or identity of species, we want to change the number of species. And what we get is a measure of areas where we have a surplus of species, that'd be in green or the green colors on this map, places where we have plenty of species, enough to get a fairly good estimate or a fairly narrow measurement error. By contrast, the orangey red dots here are showing you places where we might have to do additional sampling to capture enough species to remedy any measurement errors to get a fairly tight estimate. So I want to diverge for just a moment, and then I will turn it over to Linda. I want to briefly ask how air quality and climate might interact to change how lichen communities change, and presumably how the forests in which they grow may change. So we can think about something like, how could we mitigate potential climate changes by addressing air quality improvements? In other words, what trajectory might we have to take to arrive at climate resilience or to mitigate potential climate effects? So what I'm showing you here is a regression surface of the lichen air scores on the vertical axis. So just to reorient you, low scores, kind of in the purple region, are dominated by sensitive species, those that might inhabit relatively clean sites, and higher scores would inhabit relatively polluted or higher deposition areas. And because our regressions, the model is a function not only of nitrogen deposition or atmospheric deposition of nitrogen or sulfur, but also of those climate variables, we can navigate along this regression surface using different scenarios, different hypotheticals. In other words, what would be the consequence on my lichen air score if I did business as usual, right? If that meant there was no change in air quality, but climate change proceeded as expected, we would end up with a slightly different lichen community. What if we did worse? What if we increased nitrogen deposition and climate warming? We would end up somewhere up this red line. What if we did better? What if we did best and mitigated both nitrogen emissions and climate forcing greenhouse gas emissions? We could actually see improvements in that lichen air score. So this gives us the ability to ask how much air quality improvement might be required to offset any changes that we expect with climate warming or other climate changes. This concept can be paired with a slightly different concept called climate vulnerability. So with the climate vulnerability approach, we can ask, where geographically can we identify hotspots of expected climate impacts on lichen communities? For example, are we seeing some of those hotspots of climate vulnerability in places where we also see high levels of deposition of nitrogen and sulfur? So those are some of the potential questions we can ask, but I realize we're here to talk about critical loads, not necessarily climate and other influences. So with that, I will pause. I will stop presenting and turn it over to Linda. Maybe I'll turn it over to Mike if he would like to introduce her, but thanks very much for your attention.
Thanks so much, Rob. Before we move on to Linda, I figure we might as well take a break and see, are there any questions for Rob? If you want, you can raise your hand, click on the little hand button icon on the top, and then we can call on you. You can unmute and just ask your question directly. And while people are like thinking about questions, Rob, I think you touched on this, but can you talk a little bit more about like why you've chosen, or it has been chosen, a 20% decline as the critical load? like why that is what we designate as harm? And is it, and that it appears that that's the same for all the different community types that you assessed?
That's correct. I might actually reshare my screen and hopefully I don't steal Linda's thunder here. Let me actually pause for a moment. So maybe I won't share that. The selection of 20% is an arbitrary but meaningful threshold, because we can also work out what is the deposition value for a 10% decrease, for 20, for a 50% decrease. So it's actually up to the managers or whoever's applying it to establish sort of what is the target or the permissible diversity decline that they'd be open to. So in a lot of what I'm not showing you here, we do have values both for single species and for whole communities that aren't just at that 20% threshold. That's just the threshold that we've decided to stick with. And Linda can chime in if she has more to add there.
Awesome.
I think we felt that again, like I totally agree with what Rob said. You know, we felt that was something that, you know, we could be reasonably sure that there was a real decrease by that point, that we'd accounted for deposition error and measurement error in scoring the lichen communities. And so it's something that we would have a lot of confidence in. But like Rob said, it's arbitrary. You could pick a higher number or lower number if you wanted to. But that's what we thought would be the best. Higher than that, it starts to get to be quite a lot of change.
Yeah.
But lower than that, you know, maybe if you had a different combination of years deposition, or you went back to the community, you would have gotten a slightly different number. So 20% seemed like a good cutoff.
And I see we have a lot of Forest Service and National Park Service managers on this call. And you mentioned that most of this data comes from the Forest Service FIA program. So thus, most likely, the national forests are going to have plots about like the state of their lichen community within their boundaries. If there is a unit that does not have current data and we collected data from those areas, can we use these relationships to better assess the state of the lichen communities and potentially the air pollution where monitors are not?
Well, I would say yes. I mean, Rob showed a map of the sites that are on the FIA grid, but we have lots of off-grid sites as well. And we do have to do that because the FIA standard grid for the forest health monitoring is 21 kilometers, which means that small wilderness areas, for example, might not be captured. So we definitely, if we want to monitor in those smaller areas, we oftentimes do have to go in and establish plots in those wilderness areas. But we use the same protocol. So it shouldn't be a problem to score them and keep track of trends. Is that your question, actually?
Yeah, no, that's, I just
Okay.
want to relate this at this point to if, for anyone unfamiliar or like is interested in getting more data for their area.
Right. So
I would also say to that, oh, go ahead, Linda.
I was just going to make a note that the FIA sampling was on all forested lands. So there are plots on private lands, and there are lots of plots on state lands, BLM lands, Park Service lands, and so forth. So we, for example, our wilderness coverage includes 440 different wilderness areas, of which about 280 are Forest Service, and the rest are Park Service, Fish and Wildlife Service, and BLM wilderness areas. Go ahead, Rob.
No, you made one of my main points that yes, it's not just on Forest Service land, and then the other point being, any new data would only enhance the model or enrich the models that we've used. So if you think like we've already calibrated the model, and any new information would just enrich the model, but also give you information about your particular sites if they were to be added.
Thanks. Any other questions for Rob? All right, well, I guess we'll move on to Linda's part of the presentation. Linda Geiser is the National Program Leader for the U.S. Forest Service Air Resource Management Program in Washington, D.C. She initiated lichen biomonitoring programs in Alaska and the Pacific Northwest regions in the early 1990s, collaborated with FIA to establish the Joint Forest Service Forest Service Lichens and Air Quality National Database, and has contributed to critical load research for lichen and other vegetation since the 2000s. She currently serves on the CLAD Advisory Board and is the on the NADP executive team. Linda, thanks for coming, and share your screen and go ahead.
Right, I'll give it a try. Oh, now I have to get back to where we were. So, did you leave off here, Rob? I think so.
Yep, that's the one.
All right. Okay, so hopefully now you can see the full screen.
Yep.
Good. Okay, so when we first started working on critical loads, we were, really interested in calculating a critical load and using the lichen communities to pinpoint sites along a deposition gradient. So basically to evaluate air quality at sites using the lichen information. But down the road, we started thinking about, well, the community score, the air score, it's not that understandable a number to most people. And maybe we could work on some other metrics that would be more appealing to our managers in a sense, because they would answer directly the questions that managers are required to think about. And so we thought about the goals of our agency, which overlap a lot with the other federal agencies. And we thought about, okay, some of the main goals that managers have, or main responsibilities, are to conserve and promote biodiversity, to sustain ecosystem health and productivity, and to prevent extirpations of rare species or species of conservation concern. So then we can turn it to, well, what question would a manager have about air pollution relating directly to their responsibility for managing the forests? And so a question would be something like, is air pollution impacting biodiversity? Is it harming forest health or productivity? Are there any species at risk due to air pollution? So that's how we developed the other set of lichen critical loads to answer those questions. So the species richness answers the biodiversity question. And looking at the functional groups gets at the productivity and ecosystem integrity question. And then the individual species response curves and where the site is along that response curve helps us to estimate whether there's a decline in any species that are of conservation concern, or even whether they are at risk of extirpation. So this is what those critical loads look like for nitrogen and sulfur deposition. Let me see if I can get my arrow here. So basically we can talk about what deposition levels cause what degree of decline in each of these indicators. And Rob's already introduced those, so I'll just continue on to the next slide. But if, for example, a deposition, nitrogen deposition of 10.4 would pose an extreme risk to forage lichen abundance. You could expect about an 80% decline in that group. So if those were important winter forage for deer or the exclusive food of small mammals that are eaten by threatened and endangered species like the spotted owl, that could be a real problem. And indeed, when you look at the distribution of forage lichens in the eastern US versus the western US, there's a great paucity of forage lichens in the east compared to the west. And here's another way of looking at the sensitivity of these four different metrics, the species richness, and then the three different, sensitive species and forage and cyanolichen. So you can see at a glance that species richness is the least sensitive metric to increasing nitrogen deposition, even by the point of 15 kilograms of nitrogen per hectare per year, you still have about 50% of the community left. But with the cyanolichens, which are the most sensitive, by under 10 kilograms, you probably have virtually no cyanolichens left on the site. And this is a corresponding graph for sulfur deposition. Another thing that we're looking at is the risk of species extirpation and how deposition is affecting individual species that may be of concern to managers. So we have rated over 350 species. I think it's closer to 365 species, which on any particular survey site is about 93% of the species that you might find at a site. So we have very high coverage of these optima ratings or sensitivity ratings. So for example, here, the top species here, Nephroma occultum, it's a cyanolichen, very sensitive. Here's the group, mainly in the west. By 6 kilograms, you are at a very high risk of extirpating this lichen. Whereas something more tolerant, like Heterodermia leucomella, another rare and sensitive lichen, would not get to that point until 12 kilograms. So it's a way to sort of look and find out quickly which species might be impacted by air pollution. And now we'll switch to applications of lichen critical loads by the federal agencies. And I want to cover it by talking about programs and activities that we do under different categories that are guided by regulations. So we'll talk about activities under the Wilderness Act, Clean Air Act, the National Environmental Policy Act, and the National Forest Management Act. And I'll start with wilderness character monitoring. So this is an interagency national wilderness system-wide effort to assess resource condition and track the status of wilderness character at a very high level, usually up arrow, down arrow, or no change. And there are many elements that comprise wilderness character that are tracked. And within that, air is one of the elements, air quality. Within the air and water quality elements, lichen critical loads are one of the measures that are used to assess air quality. And within the Forest Service, we have a program called Wilderness Stewardship Performance. And this is a program that was designed to help our wilderness managers evaluate how well they're doing at stewarding the wilderness. And so it's more about tracking the activities of the wilderness program and showing that they are taking steps to steward the wilderness. So one of the arenas of wilderness stewardship performance is tracking the air quality element. And the wilderness areas earn points for monitoring air quality, wilderness air quality values, of which one of the wilderness air quality values is lichen critical loads. So one of the nice things that Rob, many nice things that Rob has done since he came to our program is develop this instant reporting tool. So as I mentioned earlier, you can look up about 440 different wilderness areas here and get an instant report that feeds back what did you learn from your monitoring. And that's the point at which the wilderness area can gain 10 points. So you get a couple of points for making a plan, a couple more for doing your baseline monitoring, a few more for doing trends, and then the final point set for actually reporting on it. And then this data can be used by the air specialists or the forest supervisors or forest planners to incorporate into air quality analyses. Another way that lichen critical loads have been used recently has been by the EPA. They are responsible for setting national ambient air quality standards for the criteria pollutants, including the primary, which is health-based standards, and secondary or human welfare standards for nitrogen and sulfur oxides. This is ideally an every 10-year process, and one of the steps in the process is preparing an integrated science assessment. And that is supposed to summarize what is the new science, what are the new findings since the last standards were promulgated. And a very nice thing is that this most recent integrated science assessment does include critical loads for lichens, herbs, and shrubs. One of the responsibilities of the federal land managers under the Clean Air Act is to identify and establish air quality related values, which is about identifying what are the resources that we manage that are sensitive to air pollution. Now that we have the lichen critical loads, we can offer a lichen AQRV, air quality related value, for all of the class 1 areas. Another program that the federal land managers participate in is the PSD program, which is designed to prevent significant deterioration of air quality in class 1 areas. It's a state permitting program. In this example, the staff from the Pacific Northwest region, the air staff, brought together data from monitoring visibility, monitoring precipitation chemistry, and lichen monitoring data, including lichen critical loads, to put together with the modeling information for a proposed gas-fired power plant to show that the location was a real concern for air resource conditions in the Columbia River Gorge. The little red dot here shows that it was proposed almost right on the border, the western border of the Columbia River Gorge. So together, the lichen data help to build a picture with other air quality data and make a case for the land manager's concerns. Within the NEPA environment, the Bureau of Land Management has recently begun using minimum critical loads, including lichen critical loads, in air quality analyses. And in this example, they used critical loads to calculate whether emissions from a proposed 5,000 well oil and gas project in Commerce County, Wyoming, would cause critical load exceedances in any nearby class 1 areas. So here you can see in the first column the critical load that they used for each of the class 1 areas. And actually this table is much longer. And the second column shows what the current deposition level is. The red colors highlight which areas already exceed critical loads. And then the project impact shows the additional increment from the project, which you can see here at a glance. The main area that would be impacted would be the Badlands National Park. However, if you added 0.022 to 3.47, you would still be under the critical load here. In the case of sulfur, there were no impacts. So anyway, it's a quick way to look at what ecosystem, whether ecosystem critical loads would be exceeded from a project. You could then delve, more into multiple critical loads if you wanted to. In this last example, the Forest Service planning directives, the 2012 planning directives, provide guidance to national forest and grassland managers about how to write a forest plan, which basically is the document that oversees the activities that occur on national forest and grasslands. And the 2012 planning directives include a recommendation to consider critical loads where they're relevant and available. And to do so, air staff will first look at deposition and assess that, identify relevant critical loads that are available for the area, and then assess exceedances. So this is an excerpt from a region-wide information document that was prepared by the Intermountain Region Air Staff to show how effects current status of air quality and impacts on critical loads of trees, surface water, and lichens. This map shows the exceedant, and they did that for all 12 national forests in the region. This is a map of the Bridger-Teton National Forest that shows where lichen species critical loads are exceeded. That's the sort of peachy areas on this map. And wrapping up here, we wanted to share with you some links to some useful tools that are already available, all of which are still evolving. And every year we're able to add more information and refine these tools. And one of the things that those of us in CLAD are particularly excited about right now is that we are developing plans to prepare an air quality dashboard for critical loads so that anyone that's interested in using critical loads can be guided through a decision tree to choose the appropriate tool to get the information that they need. So last slide. Thank you all for listening, and we would like to acknowledge our contributors to these figures, and also to the folks that have given feedback on these figures over time. And it's really, I'm just so pleased that we've been able to take this time to share with you this long story from the collection of the data since the late 1980s to the present day, where we have all of this information and tools that we can share with you. So with that, I am finished, Mike, and Rob and I can take any additional questions.
Thank you so much. Are there any questions for Linda or for Rob? Feel free to just raise your hand, and we'll call on you.
So
Never?
Oh, okay. Yeah. Hi, Mike. I was thinking, I thought I clicked my hand on, but I wasn't sure. So that was really great. Thank you, Linda and Rob. And so I don't know, maybe this question might be more for Rob, but I was really interested in one of your last slides when you were kind of talking about the effects of climate change in combination with air pollutants and how this might be, you know, the synergistic effects of those two combined. And so I was just kind of curious if there is research going on, or if there are some studies where they've looked at both of those for some of the mammals. Like I think you were talking about, they're starting to look at it for the lichen species themselves, but then sort of taking it that next step and thinking how that might be affecting some of these mammals that might be eating the lichens, if there's any work like that going on.
Wow, I like where you're going with that. I'm not aware of any. Maybe Linda is, but I'm sure there's someone out there that I'm unaware of. Linda, have you heard of anything like that?
So I need to clarify the question. So do you mean effects on animals related to changes in the lichens due to climate vulnerability, or some kind of a similar analysis for animals like Rob showed for the lichens?
Yeah, so I think the latter. Like I was thinking about pikas, which I know eat lichens and are affected directly by climate change. And then I think caribou as well. You know, they're also sensitive physiologically to what's going on with climate change, and I believe they eat lichens. So I was sort of trying to think about, you know, things like that, where it's almost like it's this 1-2 punch of whatever might be happening with pollutants and climate change acting synergistically, and how that might be affecting some of these. And I guess it doesn't have to be mammals. It could be birds or really any wildlife, you know, just thinking along those lines.
Oh. Well, in terms of a nationwide effort, no, but there are individual studies, you know, in specific geographic places that tie lichen decline to changes in bird populations, changes in invertebrate diversity and abundance, and so forth. So those links exist, but how it would happen over the whole country, I don't know.
Okay.
We would need a wildlife database, wouldn't we? That we could somehow correlate.
Emmy, Quinn, do you have a question?
Yes, thanks, Rob and Linda. That was really interesting. I was wondering, with nitrogen and sulfur deposition going down in the east, what's the kind of time period for recovery for some of those more sensitive species? How quickly do you think you'll be able to detect that if it happens?
Oh, I know that is such a good question. And some of the studies that we've done recently, well, there's a very nice set of reports for the Ohio River Valley, which had some of the greatest depauperization of the lichen community because of all the coal-fired power plants in that area. And after the air quality started to improve, some of the lichen sites were revisited over about a 15-year period, and many sites went from a practical desert to a community that had mainly tolerant species and some intermediate species. So that's about 15 years. But going from there to the sensitive species, I think that's a bigger step, because a lot of the sensitive species are not necessarily very quick to disperse. And if you have a big area that's been impacted for a long time, it can take a while to get back that full diversity. And actually, Trent Wickman, I think, is on this call. We just did a revisit to some sites in Minnesota, Michigan, and Wisconsin, where we could definitely show that emissions had improved, but the lichen air scores had not really improved significantly. You could still see a very nice gradient in the scores from the cleaner sites in Minnesota to the poorer sites in Michigan, but the rate of change was not keeping up with the rate of emissions cleanup. But that's not to say that it's just a lag. I do believe that eventually the scores would go back up.
Yeah, that's a great. That's what I was thinking about all through this presentation, is that concept of recovery. I know it's a different story in the East and the West maybe, but that whole issue of recovery and how long is the recovery and what are the pieces of that? Is that a biogeochemical cycle? Is it a dispersal biological thing, or is it a combination? I think that's going to be, in the years to come, what we got to think about.
Right, and it's a reason to continue monitoring, because there is a tendency to say, okay, we have the critical loads, we don't need to measure anything anymore, but the monitoring is what tells you, are you really getting back to where you think you should be?
And with that dispersal question, just to stick on this topic a little bit, how similar are dispersal mechanisms across lichen species? Are they similar, or does each kind of have its own method?
Oh, Rob, I know you're probably feeling the same. Isn't it wonderful to be asked these kind of questions about lichen biology? It's so fun. Well, that is a really good question. And one of the things that we actually train our field crews to notice is these reproductive characteristics and modes as ways of distinguishing species. So for example, the long beard lichens that Rob showed you in the beginning of our talk, those tend to disperse either just by dropping off big pieces of themselves, or they have tiny little structures called serratia that look like pollen grains that have both organisms in them that can disperse. But those heavy pieces tend to not go very far sideways. They tend to just drop down the tree. So those forage lichens can be among the slowest lichens for dispersing. Whereas on the other hand, if you look at some of the matrix lichens, they may have these little fingers that are actually like an extension of the lichen, or they may have the serratia, the powdery pollen-sized grains, and some of them can disperse so quickly that you might actually see them on leaves, like on an oak tree that managed to hang on all year. So they can cover the tree pretty quickly and be very responsive. So, I mean, the sad thing is that it does seem like the most ecologically critical species tend to be the slowest ones to disperse. Did you want to add to that, Rob?
No, that sums it up. I mean, I think of like there's as many dispersal syndromes in lichens as there are, there's as wide a spectrum as there is from a dandelion to an oak tree to a bamboo. Like it's a huge spectrum, and the mode of dispersal, as Linda mentioned, does impact presumable distance. And I mean, the diaspores are up in the atmosphere, like they get around, storms happen and whisk them up, but that doesn't mean they're necessarily going to establish. So a common theme both with the deposition studies and climate studies is a lag period, usually of, sub-decade or maybe on a decade scale, something like that.
Some of my favorite examples are from looking at distributions of lichens. There are polar lichens that are in the north and in the south, but not in between, that are thought to be dispersed by birds. So there are all kinds of mechanisms for dispersing lichens, but I think mostly it's wind and just gravity and rain.
Thanks. Chuck Sams, do you have a question?
Not really a question, but a comment. I was really intrigued by Jennifer's question about animals' impacts from degradation of lichen. And I know we do have some information along those lines, but I think we have an opportunity to influence the wildlife staff by educating them to a greater extent about those kind of impacts. I particularly think about the northern flying squirrel as being something that could show some effect. So yeah, I just wanted to state that. Thanks.
I guess my other thought is, I know that this was all on epiphytic macrolichen, but from my time in the Southwest, I know there are a lot of crustose lichens out that way. Have there been any kind of regular surveys of crustose lichens where we could look at deposition or climate impacts to those as well?
Mm-hmm. Well, in Europe, they have a sort of parallel protocol to the one that we use in the United States, where mostly we use the FIA protocol. And that one does include crustose lichens. And it's a much more intensive approach. We have pretty much avoided using the crustose lichens just because of the difficulty to identify them and the time it takes to identify them. You have to look at the spores, you have to make sections, and just really adds to the cost of the monitoring and also to the training level of the field crew. The FIA protocol can be used to train, summer seasonals, college students, and the crustose lichens really almost need a lichen expert to distinguish them. But the European protocol does work very well with the crustose lichens, and we even did a very nice comparison study of the US and the European method to show that they did, measured on the same sites, they did provide similar results in terms of diversity and ability to place the site on an air quality gradient.
But I think, Mike, you were mentioning soil crusts, if I'm correct. So I'm not aware of any like systematic nationwide surveying or monitoring scheme, but certainly individual researchers have done that. Academics, certainly the USGS group out of Moab. So there's been, I'm sure they have plots everywhere, but I'm not aware that there's a systematic approach in terms of monitoring within like an agency framework.
Mm-hmm. Okay, thanks. Any final questions for Robin?
So I had another question.
Yeah.
Sorry, yeah. So I had another question kind of related to that as we were talking about that. So it seems like there's a lot of conversation in the desert Southwest about the lichen, the crustose lichen that Mike was talking about, that it's in decline in a lot of areas. And so Linda, especially, it sounds like you've been out and about for decades looking at lichens across these large landscapes. Have you actually seen that, or I guess just any insight or any thoughts that you would have in terms of what you've seen? Have you noticed? Is it noticeable in some areas, or yeah?
I'll speak, and then let me let Rob speak too, because he's actually done a lot of sampling of lichens in tundra and places where the climate is changing rapidly. But I actually applied for my job here because of my concerns monitoring the lichens over 20 years in the Pacific Northwest. And in that area, the air quality is improving, as it is over most of the country for nitrogen and sulfur deposition. So what I was seeing was a change in lichen communities related to climate. And that seemed to be not even only, this fine scale climate change that you can measure using lichen communities, but just sort of a blunt force effect on the number of forest fires, the amount of acres that were burning, and the condition of the trees. So I think the climate signal I predict is going to have a bigger effect on our various ecosystem components over time, because hopefully in most places we're moving in the right direction with air quality.
Mm-hmm. Mm-hmm.
And I would just add to that, it certainly depends on which location you are, what will be the most relevant thing impacting your lichen community. So yes, perhaps we're in coastal or interior Alaska and we're concerned about climate impacts, where we're in an otherwise relatively clean air, a clean deposition site. I've done work in the Mojave Desert where I studied post-fire ecology. And so disturbances are often, going to change a community in a flash with what may have taken decades to change from climate or deposition. So as is usually the case, we can't just consider something in isolation, but yeah, often with a lot of the soil crusts, disturbance is actually the most immediate threat.
Okay.
Mm-hmm. More questions. We love questions.
There's been good ones already, yeah.
Yeah.
I have another question. This is Emmy. Can you tell us a little bit about what your training program is like for your field staff?
Sure. Within the air program, within the Forest Service air program, we use the FIA Lichen Communities Indicator Protocol. And that was developed by the Forest Health Monitoring Program in conjunction with EPA. And it's been used since the early 1990s. And basically, we have a training program, and when I used to coordinate the program in the Pacific Northwest, we'd take about a week to hire, to train seasonals, of which some of that was used for safety and navigation and how to make sure folks didn't get lost and could call for help. But a good solid 3 days, if not 4 days, is devoted to first learning how to differentiate the lichens by learning the different structures and what to look for, colors, shapes, and so forth. And then several days in practicing the protocol, which is basically a 2-hour survey centered on the FIA central plot point. And the surveyor basically has 2 hours to walk around the plot, trying to find all the different habitats and collect a sample of each species that they can find. And at the end of that 2 hours, or if they go more than 15 minutes without finding a species, they can stop. And then they assign abundance ratings, which are on a logarithmic scale. So 1, you just saw a couple of them. 2, you saw up to about 10 to 15 or something like that. 3 is more than that, but not on more than half the trees. And 4 is the lichens on at least half the trees. So that's the protocol. They package that up, label it, and then it gets identified by a lichen expert. So the field person needs only to be able to differentiate species. They don't need to know the names of what they're finding. Of course, it helps to know some of the common ones because you'll see them over and over. You won't have to struggle every time you find it. But I find that people that have never done it before actually get quite good pretty quickly. And if I train somebody and they make it through the whole field season and they want to come back, they're like number 1 on my list, because that second year, people, they still are humble. And so they'll look for a lot, but they know a lot having practice already for a season. So it's really good to have a cadre of people that can come back year after year, because it's very difficult to remember how to do it if you just get the training and then only try it out once or twice after that. Try to do it again the next year.
Thank you.
Any last questions?
We are interested in, especially for the Forest Service folks here. Amanda Hardman is our national lichen coordinator, and she is interested in helping to develop a cadre of people within the air program or the forest or the wilderness program that are interested in learning the protocol that could go back, year after year to do sites. So we will train people that would like to learn.
Well, thank you very much, Linda and Rob, and thank you to everyone who showed up to listen today. This meeting was recorded, and the link will be sent out hopefully tomorrow of where you and your colleagues can download this. I did put a link in the chat. There's a quick survey just kind of saying what was the most useful components of these talks, what can we add to our future talks to make them more relevant to your current position. So if you can fill that out, only takes a minute. And hope to see you back here March 24th. We'll be talking about critical loads of nitrogen and sulfur for tree species growth and survival. So using a lot of the same national FIA data sets to assess how trees are responding to nitrogen and sulfur deposition. And thank you very much, and we'll see you next month.
Thanks, Mike. Thank you, Rob.
Thank you.